All right, today we're diving into a full-on scientific detective story. Our mission? To find a tiny, almost invisible molecule that, believe it or not, holds immense power over our health.
So get this, right now, inside the trillions of cells that make you, there's a constant, silent struggle going on. It's this microscopic power play that determines just about everything, from how we grow to whether we get sick. It's a battle for genetic control.
And the winners of this cellular battle? They're not the biggest. They're not the strongest. Nope. They are some of the smallest molecules known to science. They're these master regulators, kind of like dimmer switches for our genes. And for the longest time, just finding them was a massive challenge.
So let's go ahead and meet the main character in this biological mystery, the mighty microRNA.
Okay, so a microRNA, or miRNA for short, is an incredibly small piece of genetic material. But seriously, don't let its size fool you. Its job isn't to build proteins itself. Instead, it's all about controlling other genes, turning their volume up or down. This regulatory power makes it absolutely crucial for development, and when things go wrong, it's a key player in diseases like cancer and heart conditions.
Now, our cells use this really elaborate assembly line to create these tiny regulators. The whole process, from a long genetic script to a final active mRNA, is just a masterpiece of biological engineering. And the final product of this whole factory line? That's the one tiny molecule we're looking for.
Which, of course, brings us to the central problem for scientists. Finding that one little active molecule is, well, it's like searching for a single specific needle in a giant, super complicated haystack.
So yeah, here's the big question. How do you actually do it? How in the world do you pinpoint the exact 22 nucleotide sequence that becomes the active miRNA when it starts out as just a tiny part of a much larger molecule?
Let's trace this journey real quick. It all starts with a long primary transcript. Then, enzymes come in and chop it into this hairpin shape called a pre-microRNA. This hairpin gets kicked out of the nucleus, where another enzyme, dicer, cuts it again. Now, this creates a two-stranded duplex, but, and this is key, only one of those strands is the chosen one. That's the active, mature mRNA. The other one, it just gets tossed aside. Our entire investigation is about finding that one chosen strand.
Now, I know what you're thinking. Can't we just find them in a lab? Well, it's not quite that simple. Traditional lab methods are incredibly slow and really expensive. And new technologies like deep sequencing, while super powerful, create what you can only call a data tsunami. We're talking billions of short genetic reads that are a total nightmare to sort through. We needed a better detective.
And so, scientists turned to computers. The first generation of these computational tools were kind of like old-school genealogists, searching for family connections.
This is what's called the homology-based approach. And the logic is pretty simple. If we know what a mouse mRNA looks like, we can scan the human genome for something that looks very similar. It's basically like looking for known relatives across the tree of life. And this method, it's great for finding miRNAs that haven't preserved by evolution across different species. But what's its biggest weakness? Oh, it's a big one. It is completely blind to brand new, species-specific miRNAs. It just can't find a suspect that has no known relatives.
So, to find those unique culprits, we needed a major upgrade for our detectives. It was time to bring in the AI-powered criminal profilers. And the difference between these methods is it's stark. The old homology approach, that's like searching a family tree. Machine learning is a whole different ballgame. It doesn't look for a specific known sequence. Instead, it learns the characteristics, the features of a real miRNA. It creates a profile of what a real one looks like, how it folds, and how stable it is. And that lets it spot brand new suspects nobody has ever seen before.
So what exactly does this AI profiler look for? Well, it learns to recognize a whole combination of features. It looks at the sequence itself. Are certain genetic letters more common? It looks at the structure. Does it form a nice, stable hairpin loop? It even looks at thermodynamics, like how much energy does it take to hold this shape together. It's the combination of all these little clues that leads to an identification.
Now, this big shift in strategy from genealogy to profiling, it didn't happen overnight. Oh no. This is an investigation that's been evolving for more than 15 years. Let's take a look at the case files. This timeline, which comes right from the source material, shows this just explosion of new tools. I mean, starting around 2004, you can see this rapid, consistent development of new software, each one trying to be a better, smarter detective than the last. This field is moving incredibly fast.
And if we look a little closer here, a really clear pattern emerges. The earliest tool, miRCheck, used a pretty simple set of rules. But by 2010, tools were already using early machine learning. And then, by the late 2010s, these highly sophisticated machine learning approaches, things like random forest and support vector machines, they became the standard. The detectives were getting much, much smarter.
So, with all these advanced AI tools, you'd think the case is closed, right? Well, not quite. There are still some pretty major mysteries left to solve. Here's the big problem. Even the best AI profilers can be a little overzealous. The source material points out that a lot of these tools generate an impractical amount of false positives. Basically, they identify too many innocent lookalikes, which then sends lab researchers on these wild goose chases, wasting tons of time and resources.
In fact, the authors of our source paper are openly skeptical about it. They say, look, when all these different tools claim to be the best, it's really hard to verify because everyone is using different evidence to test them. So they're calling for a standardized data set, a common set of crime scenes, if you will, so we can finally, truly compare which computational detective is the most accurate.
And that leads us to the final, really fascinating question the researchers pose. As we get better and better at this, can we move beyond just a general profile and find a unique genetic fingerprint for the mRNAs in every single species? Could we one day instantly tell the difference between, say, a human mRNA and a plant mRNA just by its signature? The investigation, my friends, continues. Thanks for tuning in.